Hello students, hope you all are doing good. I'm Nitin and today we will understand the insertion of transmembrane proteins and protein folding in endoplasmic reticulum. This is in continuation with session 21. Well, there are four ways by which insertion of transmembrane proteins takes place in endoplasmic reticulum. Let's talk of the first case, when cleavable signal sequence is present at the N-terminal. The N-terminal of polypeptide chain enters the lumen and the signal sequence is cleaved by signal peptidase. When transmembrane sequence arrive, translocon recognize the sequence and allow the protein to exit the translocon laterally and anchored in endoplasmic reticulum. The translation continues and the C-terminal lies in the cytosolic region as shown in this figure. In the second case, there are two scenarios. In first scenario, when signal sequence is not present, N-terminal of polypeptide chain lies on the cytosolic side and protein enters the lumen and C-terminal lies in the lumen side. However, it may happen that N-terminal enters the lumen and C-terminal lies on cytosolic side as shown in the picture. In the third case, insertion of multipass transmembrane proteins occurs. The N-terminal lies outside and many transmembrane signal sequence are encountered, because of which loops are formed with multipass transmembrane protein development. In the fourth case, transmembrane sequence lies on the C-terminal which are not recognized by signal recognition particle. This type of proteins are recognized by TRC40, a targeting factor, after completion of translation. The TRC40 brings this protein to GET1, GET2 receptor present on endoplasmic reticulum membrane, through which insertion of these proteins occur in endoplasmic reticulum. The C-terminal in these proteins lies on the cytosolic side. Now let's understand the protein folding and processing in the endoplasmic reticulum. The HSP70 chaperone and BIP protein is thought to bind to the unfolded polypeptide chain as it crosses the membrane and then mediate protein folding within the endoplasmic reticulum, as shown in this figure. The formation of disulfide bond between the side chains of cysteine residues is an important aspect of protein folding and assembly within the endoplasmic reticulum. In the endoplasmic reticulum, because of oxidizing environment, it promotes disulfide bond formation facilitated by the enzyme protein disulfide isomerase. Further, protein glycosylation also occur within the endoplasmic reticulum by the addition of a 14 sugar oligosaccharide to an acceptor asparagine residue as indicated with ASN in the picture. The oligosaccharide is synthesized on a lipid carrier called dolicol, anchored in the endoplasmic reticulum membrane. It is then transferred as a unit to acceptor asparagine. Further, 3 glucose residues are removed while the protein is still within the endoplasmic reticulum. Well, this is all about insertion of transmembrane protein and protein folding in endoplasmic reticulum. I hope this video would be helpful to you all. Thank you so much, Archana ma'am, for all the support and motivation in the development of cell biology video series. Many thanks to all of you for appreciating our videos. Please subscribe to our channel, Mentorship Towards Sure Abundance. Keep learning and stay healthy. Thank you so much.